While you share your screen, I start making an introduction here. So Blanca was one of my students here when we have the lifestyle management or lifestyle medicine MPH. This MPH ended up merging with health education, and then we have health education and wellness. But she was one of the great students with us, and then we met together in Bolivia last year doing a conference, and she presented the topic on epigenetics and other topics, and I felt that we need to have these applications for preventive care, and that's why we invited her to come today. Blanca, this is your time. She is a physician and she is a diplomate in the American College of Lifestyle Medicine and also a health coach. So has all the credentials that are needed.
Thank you, Dr. Dos Santos. For me, it's a real privilege just to share a little bit of my knowledge and my passion, which is epigenetics and another topic related with lifestyle. I really appreciate your invitation, and I felt so happy to see you all, and especially Dr. Dos Santos, Dr. Medina, who was also my teacher. Hopefully this lecture can help you to go deeper in epigenetics and learn more about that and apply not only for your practice, but also for your daily life. Let's start now then. Something is happening here. Doesn't want to go. Technology sometimes is... Okay, let's start with the definition of epigenetics, which is the study of how your behaviors and environment can cause changes that affect the way your genes work, but not change the order of your genes, the code of your genes, how your genes are coded. Unlike genetic changes, epigenetic changes are reversible and do not change at all your DNA sequence, but they can change your body, how your body reads this DNA sequence. Let's just present something related with history. Conrad Waddington, this British biologist during the 1940s, he coined the term epigenetics to describe inherited changes in phenotype without changes in the genotype. This is very interesting because here is when we can see what the man can manipulate only, the epigenetics. Let's go. Some research related with epigenetics, the most recent is with Dr. Ornish, one of the big programs here in California, who is related with heart disease reversal program. He already has a CN in Sausalito, and also the prostate cancer. He found that after changing lifestyle behaviors in his patients, they found that more than 500 genes showed alteration in their expression. Also, the EPIC study designed to investigate the relation between cancer and nutrition. The glucocorticoid receptor studies, which related with the stress regulation. This was made in rats. So they lick their little babies, and those who didn't receive this licking from their moms presenting after when they reach the adult age, some unregulation related with the stress. The Dutch hunger study that most likely you already know, those moms who were pregnant during the war in Netherlands when were occupied by Germany. So they give born babies with some alterations in epigenetics, and when they reach the adult stage, they had a lot of problems related with chronic diseases. The agouti gene, also in a study in mice, these rats who were exposed to BPA, they give birth to babies who were in a different color, their skin, especially their hair, who were yellow ones. So these are some of the very popular studies related with epigenetics. Epigenetics is related to gene expression. This is active genes versus inactive genes. It's like a turn it on and off some of the genes without changes in the DNA sequence. Epigenetics is needed for normal life. All our cells need to be activated or deactivated in some specific genes to become some specific cells too. For example, all the cells came from stem cells, but to become a neuron, some genes have to be activated or deactivated. Same happened with all other kind of cells. Enterocytes to become intestine cells, hepatocytes to become liver cells, cardiac cells, etc., etc. Some interesting facts related with epigenetics and genetics. I mentioned now, it will take nearly 5,000 strands of DNA laid side by side to equal the width of a human hair. In average, human chromosome will be about 621 kilometers, equivalent to 385 miles long, or roughly the distance between San Francisco and LA. What amazing God we have. Also, the oldest book describing epigenetics, according with my research, is the Bible. I present these verses who are related with, mention something of trigenerational epigenetics effects in Exodus, Galatians, and Proverbs. The karyotype. This is very interesting because it's something that we cannot change, but we can alter the expression of the genes only. The karyotype is obtained during metaphase. Many of you probably know. But the most interesting thing is the silencing and activation genes to define the embryo's gender. These phenomena have been termed meiotic sex chromosome inactivation, also known as MSCI. This is an epigenetic silencing process that occurs exclusively in germ cells. So males are who define the gender of the baby, and it's because of silencing one of the genes. We are the most incredible and perfect designs. Each chromosome contains hundreds to thousands of genes. Genes are made up of DNA. The Human Project estimates that humans have between 20,000 and 30,000 of genes. Most genes are the same, but a small number of genes, less than 1%, are slightly different between each individual. This don't happen in the microbiota. It's very interesting because in microbiota, we have a huge difference between genes. Your genes play an important role in your health, but so do your behaviors and environment. There is a very close interaction between the environment and our genes, such as what you eat and how physically active you are. Let me present to you, this is a very brief, it's a very short video. Let me know if you can hear the sound. So less than a minute.
The sound is not there.
I'm sorry, you cannot listen the sound?
No.
Oh, let me share there. Let's see.
DNA in our bodies
Now it's okay.
is wrapped around.
Now it's okay?
DNA contains
Fine.
the instructions for building all the parts of the body. But DNA is only half the story. The DNA in our bodies is wrapped around proteins called histones. Both the DNA and histones are covered with chemical tags. This second layer of structure is called the epigenome. The epigenome shapes the physical structure of the genome. It tightly wraps inactive genes, making them unreadable. It relaxes active genes, making them easily accessible. Different sets of genes are active in different cell types. The DNA code remains fixed for life, but the epigenome is flexible. Epigenetic tags react to signals from the outside world, such as diet and stress. The epigenome adjusts specific genes in our genomic landscape in response to our rapidly changing environment.
If we understand this concept, we are just having a more clear vision of what epigenetic means. Epigenetic mechanisms had several of them, but relating with medicine, lifestyle medicine, 3 of them, mostly 2 of them, they are the most important. The DNA methylation, and we saw the little tags of methyl groups that can be attached to especially to the cytosine. The histone modification that is also related with tags can be acetylated or methylated. When it's acetylated, they favor the expression of the gene. When it's methylated, the gene is silenced, and the non-coding RNA. All this happened during the first stage of the DNA process. But this epigenetic pathway can be direct, can have a direct effect or indirect effect. When we talk about direct effect, it's related with modification of biochemical pathways, abnormal availability of epigenetics enzymes, which end up in the aberrant epigenome, and indirect effect is when this chronic exposure of altered expression of growth factor receptors, ion channels will lead to an aberrant epigenome. Methylation, which is the first mechanism that we are talking about, happened during transcription. It's one of the epigenetic signaling tools that cells use to lock genes. That means turning off position, plays an important role in embryonic development. Very important role, because here's where reprogramming gene can happen. Genomic imprinting, X chromosome inactivation, and preservation of chromosome stability. When DNA is methylated, nearby histone are deacetylated. That means that we are turning off the gene expression, resulting in one inhibitory effects on transition, on transcription. Here is an example, a very easy to understand example. DNA methylate transferase just add 1 methyl group in the position 5 of the cytosine. When this happens, the methylated DNA just turn it off, and there is no expression of the gene. The histone, they become very tight, so that cannot be expressed. And when it's unmethylated, these histones are more loose and then can be expressed, meaning the gene is turned on. Epigenetics is like maybe considered as the intersection between genetics and the environments. Modification can be influenced by environmental catalytic, such as diet, smoking, or exercise. Our behaviors matters significantly when we are talking about epigenetics. DNA methyltransferases, these enzymes, can be activating or deactivating this gene. Transfer methyl group from universal methyl donor, who can be, for example, even vitamin C, folic acid can be a methyl donor, but for health. Mostly of them, when they are methyl donors from the unhealthy environments, is resulting in some diseases. Establish, recognize, and remove DNA methylation too. When methylation happens in an aberrant way, hypermethylation and hypomethylation are the two events that can result in diseases. Hypermethylation has been linked to autism, cardiovascular disease, obesity, type 2 diabetes, and cancer. Hypomethylation, by the other way, is associated with nearly all human cancers. So, DNA methylation changes can happen related with environmental factors or biological factors. Some environmental factors are aging, lack of physical exercise, stress, depression, high or even lower alcohol consumption, air pollution, and related with biological factors in activation of the X chromosome, genomic imprinting, reprogramming of the genome during differentiation, development and survival, genetic and molecular alterations. Some of the abnormal patterns of methylation are cancer. Hypermethylation is signaling tumor suppressor genes. For example, the p53 gene in chromosome 17, the BRCA1 in breast cancer, colon cancer, and hypomethylation, turning on or activating pro-oncogenes. TNBC, which is a very aggressive breast cancer, the triple negative cancer. And when histone modifications mechanism happens, acetylation, which is when the gene can be expressed, occurs in amino acid lysine in the histone tail. Acetyl group is added to lysine, and histone acetylation leads to transcriptional activation. Another mechanism, as I mentioned before, is phosphorylation, and the other that we already mentioned too is the methylation, but we are more focused now in acetylation and methylation. Histone modification, acetylation, produce chromatin structure changes. So the chromatin becomes more loose and the gene can be expressed. It's correlated with transcriptional activation. Histone deacetylation leads to gene transcription, meaning silencing. The good news is the epigenome is changeable, can be reversible when it's aberrant. During lifetime, different genes are needed. Cells are constantly listening to signals to change what they are doing. Only imagine when you are eating something that is unhealthy, your cells are waiting for you to hear what are you doing for them. Signals come from inside the cells, which highly depend on what is happening in your environment. Everything matters. It's well known that identical twins, if they have a different lifestyle, they will have a different outcomes, a different phenotype. Matters personal choices, where we live, the society that is where we are, our environment, even the spirituality can change our epigenes. Interpersonal relationships, it is about our past, present, and future. Genes may be activated or silenced epigenetically during different stages of life, but especially during pregnancy, the fetus and the infant, they have a very vulnerable effect related with this interaction between environment and epigenes. The interaction between genetics and environment is responsible for the development and maturation of the individual through his or her life, the entire life. But the main stage when can suffer more damage is during pregnancy, what is a fetus, and infancy, because this plasticity that can present the unborn baby do not change the DNA sequence, but they can change how the body reads this DNA. Nutrition, physical exercise, any chemical compounds, pharmacological treatments, and healthy habits will affect lifelong. This is the transgenerational effect in our epigenes, because moms who are pregnant during generation 1 can affect the epigenes of this baby. And if this baby is a female, already carries these changes to their future generation. This can be affecting even for the third generation. The brain are particularly sensitive to epigenetic changes. Experience it very early during life, when the brain is developing more rapidly, cause epigenetic adaptation that influence whether, when, and how genes release their instructions for building future capabilities for health, skills, and resilience. That's why it's crucial to provide supportive and nurturing experiences for young children during the earliest years of life. Let's talk a little bit about adverse fetal and early childhood experiences, also known as ACEs. This can lead to physical and chemical changes in the brain that can last for life. Poor nutrition, exposure to chemical toxins or drugs, and harmful stress during pregnancy or in early childhood are not forgotten, but rather would impact the developing brain through the epigenome. So before, few years ago, we were believing that during the conception, during the fertilization, all the genes were like reset. Everything was erased and starting from the novel. But now we know that some epigenetic tags, can be carried through generations. The biological memories can affect multiple organ systems, increase risk for poor physical and mental impairment, and detrimental future learning capacity and behavior. Social determinants of health, which is highly related with public health, can actually impact the epigenes of people who are exposed. Having access to healthcare during pregnancy, infancy, and toddlers is a protection factor. Sorry. I'm sorry, the support for new parents and caregivers. Supportive relationships, access to education, access to good nutrition, and low psychosocial stress exposure will help individuals to lower the risk of health harm. Oh my, let me see. Let me see, it's not going to. Technology, okay. Here again. Nutrition and epigenome. This is an amazing example of how diet can impact the phenotype. Bees, the queen bees are genetically identical to worker bees apart from diet. It's the only difference. Worker bees are sterile. They are fed with royal jelly only during the first 2 days of their larval stage. Queen bees are fed with royal jelly during their entire life. That is the big difference between these two little bugs. Royal jelly results in queen developing ovaries and large abdomen for egg laying. Some bioactive dietary components in our diets that I really encourage to consume every day are the polyphenols. Those exceed more than 8,000 polyphenols different, but one who I want to mention now is the EGCG found in green tea, the curcumin found in curry, and resveratrol found in grapes, and also in pomegranates. They are protective against cancer. It has a very important role against many diseases, not only in cancer, but this is one of the main related with epigenetics. Significant impact on cancer prevention and has the ability to alter the epigenome in cancer cells, chromatin remodeling, reactivation silenced genes, especially those who protect against cancer, induction of apoptosis and cell arrest in cancer cells, inhibition of oxidative stress and angiogenesis, which is very important to relate it with cancer, and regulation of signal transduction and reduction of cancer cell proliferation. If you don't like some of this, just try to consume every day to integrate in your diet, and eventually after 2 weeks, you will like it. Calorie restriction, this is a very interesting study too, that may induce epigenetic changes that have the potential to alleviate age-related diseases and or impact gene reprogramming during pregnancy. We don't have to spend a lot of money for keep us young, right? And alcohol is associated also with harmful epigenetic modifications, produce hypermethylation of numerous genes and good numbers of genes. That means that turning on silence, develop in the development of several human cancer, like colon cancer, hepatic cancer, breast cancer, also the hypermethylation in heavy and light drinkers than compared with not drinkers also is related with neck and head cancers. So there is no safe levels of alcohol intake, 0. Physical exercise impact. This is one of my favorites, and I know Dr. Medina is very familiar with this. DNA hypomethylation and histone hyperacetylation is resulting from physical exercise. It depends on the time, intensity, and duration of the physical activity, and promotes the demethylation in brain-derived neurotrophic factor, also known as the BDNF, and increased acetylation of histone. That means that we are activating our genes. Promote the expression of mitochondrial genes, this mitochondria, which is the main source of energy in our body. Hypomethylation, early exercise response in some skeletal muscles, genes, and always will have positive health outcomes. I know that in public health, we don't, it's supposed that we don't say always, but honestly, I see physical exercise as medicine, one of the best treatment ever that we can use for ourselves, including for our patients. Improve metabolic efficiency, improve oxidative capacity, and improve contractile activity as well. The PGC1 alpha gene is one of the most popular mentioned related with physical exercise, become hypomethylated after intense exercise. Regulatory gene for mitochondrial biogenesis. If we have more mitochondria, we'll have more exercise and longevity. Fatty acid oxidation and skeletal muscle insulin sensitivity. Increased levels of mRNA 3 hours after endurance exercise. So we know, and physical exercise physiologists know, that the benefit is not only during the time of exercise. We have the benefits hours after this activity. Another gene, the TFAM, the mitochondrial transcriptor factor A, become hypomethylation after one exercise session and maintain 3 hours later. Mitochondrial DNA, important role on a stress-mediated inflammatory response and protect from reactive oxygen species and degradation, increase mitochondrial function. It is very important to recommend the preconception parental physical exercise, especially in moms, improve the unborn baby glucose tolerance, favor the body weight, healthy body weight, and C-reactive proteins profiles, which is a biomarker for inflammation. Low hippocampal DNA methylation too. Sleep deprivation, which is a very serious problem now, not only in adults, but also among youth, may alter expression of more than 700 genes in adults. This is linked for many unhealthy outcomes. Environmental influences can be the BPA exposure, which is in plastics, heavy metals, pesticides, diesel exhaust, tobacco smoke, radioactivity, hormones, nutrients, infection diseases, then were led to many different diseases after the epigenetic modification. Why we should study more and do more research related with epigenetics? Because it's useful as a biomarker of exposure and disease, targets for modification through preventive and therapeutic interventions, and create new policies based on the result that we can have after conducting epigenetic studies. And update, why not, the current health system, that we really need that. And just as a reflection, experience of life choices shape human body and mind. Gene expression is modulated by the interaction of nature and nurture. It's our own decision what we while choosing every day, what we want to drink, eat, the physical activity that we want to practice, if we're rather to ride the bike or the car, is up to us. Epigenetic transformation is possible. And remember always that DNA is not our destiny, but what we do every day in our behavior can be our final destiny. Thank you so much. I hope this can create more awareness, this lecture, and just awake the curiosity in your inside, just to start developing more studies related with epigenetics. Thank you so much for listening.
Thank you, Dr. Ponce. It was very great. Let me start with a question here. Many times when we see this type of epigenetics lecture, we get an impression that everything happens during pregnancy and infancy, and that's it. It was my mom's fault. What can I do now to counteract that?
So we can change our epigenetics. That is the beauty of the epigenetics, that we have in our hands lay the response. Do we want just to continue with the same habit, the same unhealthy behaviors that we inherited from our parents, or we want to revert these unhealthy behaviors and just like starting to turn it on or turn it off the genes that we have in our bodies? There are some less than 10% of the outcomes that health outcomes that we cannot just manipulate or we cannot change because they are linked directly to our genes. But we have a 90% and probably more because the epigenetics is evolving rapidly nowadays. So probably more than 90% that we can correct and we can switch, just changing our mindset for more healthy behaviors.
Yeah, I guess.
Yes, Dr. Medina.
Just a little bit more. I guess when
Yes.
we saw this study by Dr. Ornish that he has oncogene suppressed and suppressor genes activated, they might get this since they were born, and now because of their lifestyle, the things switch. The problem is that we always get our minds fixed on what happened during pregnancy, and that's too late for me, and then I have to teach the next generation. But it's not too late for us to change. So that's the point. Okay,
Yes.
other people asking questions. Sorry.
Yeah, Blanca, very, very good. I really enjoyed your presentation. Nice to see you again.
Nice to see you, Doctor.
And I wanted to comment on, I wanted to just ask a little bit more about that last statement you said. You said over 90%. I've seen different percentages, at different meetings and conferences, 75, 80%, is due to behavior and environment as far as being able to impact or make some change in your gene. Do you have any like a specific reference? Because I've tried to actually find a reference that says that you're 90%. Do you have
in one of the, I'm sorry, Doctor. Just finish.
Okay.
Go ahead.
Go ahead.
Yeah, I can look and share with you the specific reference, but in the reference that I presented at the very end, in several of the articles, you will find that. And also I can't, because it's something that state the American College of Lifestyle Medicine, I can share the data too.
Okay.
With you.
All right,
Yeah.
great. Thanks.
Yeah, and something very interesting is that 70% of the regular doctor visits are related with lifestyle diseases that can be prevented. Even a good number, I don't have the specific number of how many of each, I think it's between 7 or 8 of 10 patients who are drinking anti-diabetic medicines. They can stop drinking that and they can revert the disease if they just change habits, like a change in diet, starting physical activity. So I don't have the number exact, but it's a high percentage too.
Thank you.
Mm-hmm. Comments?
Hi.
Questions?
Hi, Dr. Blanca.
Yes.
¿Cómo estás?
Hi, muy bien.
Good to see you.
Good to see you too.
Just to, how do you apply the text that you mentioned in the Bible to epigenetics? What are you trying to say with those texts?
Okay, with those texts, I especially Exodus, if you can see there, that say that that happened, the sin of the parents, well, be reflected until the third generation. You know that sometimes it's hard to say, but habits sometimes it's because resulting from our sins, for our behaviors, unhealthy behaviors. So for intemperance, you know, so, and this intemperance can go transgenerational. So our grandkids can suffer my love for eating a lot of candies, my unhealthy behaviors. They may suffer the consequence of this through diseases. So I don't know if I answer your question, or probably you have a very view of this, because I know that you are familiar too with this topic.
Thank you, Lupita. No, that's what I wanted to see how you reply.
Maribel.
Yes.
Hi, Dr. Dos Santos. This is Maribel. Hi, Dr. Ponce. Very good presentation, very informative.
Thank you.
Yeah, I just have a very simple question. I posted through the chat, but I think it wasn't obvious. Anyway, so my question is, so we know that genes are inherited, but can once we make those epigenetic changes, can they also be inherited as well? I mean, I don't know if that's
they can, yeah. So they don't change the genetic code. So the genetic code is a combination of these 4 nitrogen base. So they don't change this, but we can get from our grandma or mom these little tags, these little acetylation or methyl groups that they can add it in the cytosine island, cytosine and guanine island. So this that can result in some diseases, we can carry this from our ancestors.
Can I comment on that?
Yes, of course.
Okay, Colin T. Campbell, you know, this great researcher, you know, he was sent to Philippines because he was a veterinarian. He is a veterinarian by career. He found out that the children were from mid-class were having hepatocarcinomas that mostly are found in adults. So they found out by the time he was studying that there was a published paper in India where they were doing a research in rats, and they realized that the animal protein was modifying the behavior of the rats. Actually, that was the production of more hepatocarcinomas in rats, and that was basically what was that? Lactose that we're receiving. And after that, they took away the milk protein, and then the hepatocarcinoma, you know, receded. So that was a very strong call to study the epigenetics surrounding these issues. And then they realized that the genes had like a switch, and that was mostly related to the foods they ate. So you can open a switch with certain foods or turn it off, and that happens with especially those genes that we have that are called oncogenes. If we eat the right foods, they will be close. They will not be manifested in disease. But if we eat the right foods, that means the strong influence on diet, on our epigenetics. And that also tells us about the Dutch hunger during the World War II. So we have a strong evidence that the epigenetics is strongly influenced in our in our lives in different aspects.
Thank you, Doctor.
I have another question that it might be controversial here, but some people, some scientists are using epigenetics to explain gay behavior, telling that the genetics can alter the chromosomes. Do you have any comment on that?
It's a very interesting question, doctor. And Dr. Pawlowski, I forgot his last name, but I know Dr. Medina knows his, it's from Stanford University. He's a neurologist. He mentioned something about that in one of his lectures, that something happened in the rewarded system that changed some expression of the genes that then they can manifest like the desire for the same sex. They have attraction for the same sex, but I don't see, I haven't read any study that states as a yes, but we don't know. We have a lot of added, substances in what we eat. I have no idea if it's related with this behavior. I don't know if somebody has any comment about this. Pawlowski, I think.
Pawlowski, yeah. Well, I don't want to put you in the spot. I just, I was discussing with a physician, and he was pushing that now we have a genetic explanation for the behavior. Well, this could be because sometimes our lifestyle can lead to alterations there. I don't know. I mean,
I think there are many factors that can contribute to that.
Dr. De Santos and Blanca, there is a study by William Rice on the title of the study is Homosexuality as a Consequence of Epigenetically Canalized Sexual Development. And that's an interesting article to kind of lead us on this type of question.
Will be interesting to know and to research much about that.
I do have another question. Are you aware of any illnesses that doctors are already like turning off or turning on genes to treat just by doing that?
Yes. One of the most popular that you can see the reverse, and I had experience in several of my patients, is diabetes. So it's incredible. It is incredible how after 3 months applying the coaching technique that I got from Loma Linda University, so they can revert the diabetes. After 1 year, all the biomarkers become normal. If they follow, if you help them just to change the behaviors and modifying diet and increasing physical exercise, helping them how to manage stress, it's very interesting. After starting with the H1C in 13.7, after a year, you have an A1C 5.5, 5.6. It's incredible, but it's possible. I think it's one of the most popular. And also a fatty liver, the change is dramatic too after they change their behavior. Hypertension. So I think most of the chronic diseases, if apply for these epigenetic changes, if they switch for a different lifestyle.
But we don't have many studies with genes on that. So after Ornish, I mean, it looks like we didn't have much. I wonder what happened to other types of cancers, if they can see what Ornish saw on the phenotype change of the gene, suppressor gene and oncogenes. I wish we have more of those, because one time I read a study in diabetics that they compare the effect of lifestyle with diabetes that had a gene for diabetes compared to the ones that didn't have the gene. And the ones with the gene were the ones that changed, that improved more in insulin sensitivity and and all the biomarkers than the ones that didn't have the gene. But we haven't seen much of these studies being populated more. I guess it's too expensive.
Yeah, most likely, and all the requirements for this kind of experimental study. But it's a great opportunity for Loma Linda.
Yeah, but where the money is in the pharma, the big pharma. So
that's true.
So now we have to be experts in fundraising and grant writing.
Yeah.
Grant writing is the key.
A challenge for our students here.
Yeah, well, thank you, Dr. Ponce, for answering the next question as well. Very impressive studies.
Thank you.
Any other questions?
But Dr. Santos, I think the China study offers an ample ways to see how epigenetics influences cancer in several areas. China study was done in 250 provinces of China.
Yeah, but
the influence.
What I'm talking about is measuring biologically the genes and not the lifestyle. The lifestyle is okay. We have enough evidences. And by the way, the China study is full of points that the researchers don't like to mention. Nowadays, they have a lot of criticizing for the Chinese study. But even if we have, I mean, what we need is to have the methylation studies. That's what we need.
Yeah, you're right. Because there is one that hypermethylation after drinking a lot of folate supplements could be related with colon cancer. So it's very interesting because we think that we find everything, but when in some way humans bring something else like supplementing for anti-cancer supplementing, then another problem just show up.
With supplements.
Nothing is better.
Supplements.
Yes. So nothing is better than eating healthy every day, doing physical exercise, managing stress, and sleep deprivation prevention. So all these kind of behaviors can help us to live healthier and happier.
Well, good discussion. Thank you very much again, Dr. Ponce, and we will keep in touch, and maybe we'll invite you for other sessions to bring.
Thank you, Doctor.
Those issues.
My privilege.
Yeah. Okay, we are gonna adjourn now. Thank you very much for everyone. And we will have this lecture recorded, so you can tell your colleagues that we will put that in our YouTube channel. Thank you very much.
Okay. Thank you. Good to see you all.
Thank you.
Bye-bye.
Bye.
Bye.